Dear colleagues, I'm very glad to see you all, and I'm very happy to present our today's speaker, Natalia Nosova, who will talk about the results of her collaborative study on cupule-bearing cones of Jarudia. Natalia, please.
Okay. I am glad to see my co-authors, and I hope for their help in answering some questions, maybe. I'm starting. The genus Jarudia, with type species Jarudia joy, was established by Gunle with co-authors for silicified seed cones with lateral cupule-bearing structures from the Low Cretaceous of eastern Inner Mongolia in northeast China. These cones consist of main axis here with helically arranged bract cupule axis complexes. Each of these complexes terminates in a single incurved cupule. Cupule-bearing stalk is thin proximally here, where it is fused with a bract. Above, it is free from the bract here and gradually widened toward the tip. It is curved adaxially relative to the main axis. These two pictures show longitudinal and plan view diagrams of the seed-bearing units. Each cupule is formed by a wide flattened distal part of the cupule stalk, this one and this one median here, and two lateral flaps. The flattened cupule stalk and three flaps are fused at the apex of the cupule, but they are free toward the base. The cupule contains two seeds, this one, that are triangular in the transverse section. A micropyle of the seed is oriented back toward the base of the cupule stalk. The seeds are erect, sessile, and borne near the apex of the curved distal portion of the cupule stalk. There are two vascular bundles near the base of the bract cupule stalk complex here. The adaxial vascular bundle supplies the cupule stalk, while the abaxial bundle supplies the bract. Distally, close to the point where the bract separates from the axis, the adaxial bundle divides into two separate vascular bundles, here and this one. These two bundles continue distally into the cupule to supply the two seeds. Recently, one more species of Jarudia was described based on a study of the material from the Aptian Albian Balaktak Formation of Kotelny Island in the New Siberian Islands in Russian Arctic. 4 seed cones and several dispersed cupules have been found in the material from Kotelny Island. This material was given to me by Alexei Herman, and I am very grateful to him for this. Initially, Alexei identified these cones as Carcania. It was published in these 2 years. Also, I thank Ksenia Domukatskaya, who permitted me to work with the collection from Kotelny Island and helped with some stratigraphic questions. We have studied these specimens using micro-CT. It was surprising when we discovered another cone inside the sample matrix invisible on the surface, this cone. Just a minute. This one. this is the same specimen with two cones, this one and this one. The cones are elongated, cylindrical, and gradually tapered toward the apex. Each consists of a straight main axis bearing lateral bract cupule axis complexes. The most complete specimens show more than 56 bract cupule axes. Each bract cupule complex bears a single cupule at the tip that is curved back toward the base of the cupule stalk. Maceration of the bract cupule stalk revealed a cuticle fragment composed of two cuticle layers. The central dark region in this fragment appears to indicate the position of the stalk, like this. And this region flanked by two wings created by the fused bract, like here. Stomata occur on only one of the two cuticular layers. In this, probably the abaxial cuticle of the bract. Stomata are arranged into stomatal bands. In transverse section, cupule stalks are rhomboidal in outline and slightly flattened. Here it is visible the free part of the bract with an acute apex. In median transverse section, cupules are rectangular in outline. Each cupule is formed by the distal white flattened part of the cupule stalk. Here is good visible. That is curved distally. Also one median, this one, here, and two lateral flaps, this one. In some cupules, there is a short apical protrusion here, and here it's visible. Here it is visible that cupule has an outer and inner cuticles, and this one visible. In micro-CT scans, the cupules appear empty, but maceration of several fragments of carbonaceous material extracted from the cupules revealed remains of the megaspore membrane, and very thin cuticle, presumably of the nucellus cuticle. The megaspore membrane is made up of a patent layer and the underlying foot layer. The patent layer is composed of crowded bacula. The bacula branch and interconnect in their basal part to form a network and often have unbranched distal ends. Maceration of the cupules revealed several fragments of the outer cuticle. Cells near the distal part bears distinct papillae, here and this one, and occasional trichome here. Stomata are rare here, with stomata, this and here. The stomatal complexes are haplotylic and incompletely amphicyclic. The subsidiary cells of some stomata bear small papillae. Here it's visible. Thus, the specimens from Kotelny Island provide new information on epidermal features of the cupules and on the structure of the seed megaspore membrane that has not previously been available. So in Jarudia joey, detailed structure of the cupule, cuticle, and seed has not been studied. Cupule-bearing structures of Jarudia borealis are closely associated with numerous fragments of linear parallel vined leaves, here and this one. This is the same sample from both sides. And you can see here the same leaves like here. Similar fragments of parallel vined leaves were found in another specimen from the same plant-bearing class. On the opposite side of this specimen, we have found a bundle of leaves with the same morphology. We studied the epidermal structure of both detached leaf fragments and the leaves gathered in a bundle and found similar epidermal features. Morphological and epidermal characters of these leaves fit the diagnosis of phenocopsis. Based on the epidermal features, we described these leaves as a new species, Phenocopsis arcticus. There are 8 to 11 veins across the width of each leaf in its widest part. The leaves are hypostomatic. Each stomata band has 2 to 4 stomata per width. The stomata are oriented longitudinally. Every cell on the adaxial surface has 1 to 6 small papillae. Sometimes papillae are fused into a cuticular ridge. Most cells of the abaxial epidermis, here and here, have a prominent longitudinal cuticle ridge. This is adaxial surface. In cells of the stomatal rows, here, papillae are also present in addition to the longitudinal cuticular ridge, and most of the subsidiary cells bear papillae here and here. The gut cells are narrow. The specimens with Jarudia contains numerous leaf fragments of Phenocopsis and rare fragments of Pitiophilum, red rose here and this one, and Ginkgoides, this leaf of Ginkgoides and this fragment. In the collection from the same locality, there are leaves of several species of Ginkgoides, Phenobira, Phenocopsis, Chikonovskaya, Exostrobus, and Leptostrobus. The structure of the stomata from the cupules of Jarudia borealis shows some similarity to the stomata of Phenocopsis arcticus from this one. Based on their close association and some cuticle similarities, it is possible to assume that the cupules of Jarudia borealis and the leaves of Phenocopsis arcticus were produced by the same kind of plant. However, the situation is complicated by the presence of Phenobira leaves in the same collection. The cuticles of these Phenobira leaves are not well preserved, and hence comparison with the cuticle of the cupules is not possible. Also significant that in many known localities, leaves of Phenocopsis as well as Chikonovskaya, this is Phenocopsis, this is Chikonovskaya, are associated with seed-bearing structures of Leptostrobus. Seed-bearing structures of Leptostrobus consists of an axis bearing spirally arranged bivalved flattened capsula. Here, one capsule. Each capsule in which ovules in these capsules are arranged in a single row along the margin. The epidermal features of the basal scale leaves here, as well as the outer epidermis of the capsules of Leptostrobus are very, very similar to those of Phenocopsis and Chikonovskaya leaves, including the scale leaves that cover the short shoots to which the leaf bundles are attached. Therefore, it is difficult to say with certainty whether the leaves of Phenocopsis arcticus and the cones of Jarudia borealis belong to the same plant. Another find of Jarudia comes from the Aption from the obtained slab formation of the Slab River basin in the Zhiryanka Coal Basin, northeast Russia. We have two specimens, are part and counterpart of the same cone. This cone consists of a main axis with helically arranged bract cupule axis complexes, each with a single incurved cupule. In median transverse section, cuples are rectangular in outline. Each cupule is formed by the distal white flattened part of the cupule stalk that is curved distally, one median and two lateral flaps. These samples were studied using micro-CT. Here it is visible the long cone with numerous bract cupule axis complexes. The cone has a thick axis. Some cuples show tooth-like conical projection on their surface. Here it's visible and here. The specimens with Jarudia species contain fragments of linear parallel vined leaves, visible this one and here. It should be noted that the cupule-bearing cones of Jarudia joy also were found in association with unnamed multi-vined leaves. The specimens of Jarudia species from the Slab River basin are still being studied. The organization of the seed cones of Jarudia borealis from the New Siberian Islands and Jarudia species from the Zhiryanka Coal Basin are directly comparable to that of Jarudia joy type species. In Jarudia joy, the cupule contains two seeds. They are triangular in transverse section. The cupule is quadrangular in cross section. In micro-CT scans, the cupules of both Jarudia borealis and Jarudia species appear empty, but the quadrangular cross-section of their cupules suggests that they also bear two triangular seeds. Jarudia borealis differs from Jarudia joy in having cones with more densely arranged lateral seed-bearing units. The cupule stalks are also thinner, often longer, and bear slightly larger cupules. Jarudia borealis also has cupule stalks that are rhomboidal in transverse section. I think that the same situation in Jarudia species here. But Jarudia joy have oval, maybe broadly elliptical stalk in the transverse section. Jarudia borealis has trichomes and papillae on margins of flaps, which have not been observed in Jarudia joy. Unlike Jarudia borealis, Jarudia joy and Jarudia species have a distinct lip at the tip of the median flap here and this one. Also, they have both tooth-like conical projection on the surface of the cupule. Jarudia species differs from other species in the shorter length of the bract cupule complexes and much smaller cuples. Here is a comparative table of Jarudia species showing sizes of cones, axes, cuples, the presence or absence of the tooth-like projections on the cupule surface, and also localities and age. As I already said, the seed cones from the New Siberian Islands were previously determined as Carcania. This is understandable since a bract cupule axis complex that bears only a single cupule, as in Jarudia, can resemble a pedicel bearing an incurved ovule as described for Carcania. For example, seed-bearing structures from the Low Jurassic of the northern Iran were originally identified as Carcania, but were recently restudied and assigned to Zerabia. I will talk about this genus some later. Similarly, Jarudia borealis resembles Carcania archangiskeliana, described from the Aptian Albion of central Mongolia. It resembles in having a cylindrical cone-like structure with a central axis that bears helically arranged seed-bearing units. Having discovered some similarities, we decided to restudy some cones of Carcania archangelskiana using micro-CT. Here you can see some photos, micro-CT photos. We did not find any cupules and bracts here. These structures look like as a pedicel with an ovule. When I started preparing this report, I remembered that Evgenia Bugdaeva once a report on cones similar to Jarudia. I have written to her about it, and she sent me some photos of specimens that resembled Jarudia cones. These specimens come from several early Cretaceous localities in Transbaikal region in Russia. As you can see, these cone-like structures have a central axis which helically arranged seed-bearing units. This one. Overall, they are similar to both Jarudia and Carcania. One of the main structures we need to find in Jarudia is the bract. As for me, it seems to me that here is bract. Also, I think that this structure is similar to a cupule. But for accurate identification, it would be good to use tomography. And now about the systematic position of Jarudia. In 1909, based on the structure and anatomy of individual cupules, Stokke and Rothwell described a new genus Doyleales, with the type species Doyleales tetrahedrosperma, from the Low Cretaceous of Vancouver Island in Canada. These authors referred Doyleales to the Corystosperms based on the structure and anatomy of only of cupules. In 2016, Rothwell and Stokke described a new find of Doyleales tetrahedrosperma preserved as a cone with cupules, not only cupules. In addition, they transferred cones of Umcomasia mongolica to Doyleales mongolica. Umcomasia mongolica was described by Gunleshi with co-authors from the Low Cretaceous of central Mongolia. Rothwell and Stokke found that the cupule of Doyleales is constructed of a vascularized petiole and unvascularized lamina lobes, whereas the cupule of Corystospermalis consists of an vascularized unilobed or bilobed lamina on recurved vascularized petiole. Rothwell and Stokke established a new order, Doyleales. Here you can see an original diagnosis. Seed cones of Doyleales have a central axis from which bract cupule axis complex diverge in a helical arrangement. Each complex consists of a basal stalk from which a bract tip separates and two sporophylls, each bearing a single inverted seed. Major seeds are surrounded by a seed-enclosing cupule of sporophyll origin. Vascular bundles terminate at the base of the seed and does not extend into the lobes of the cupule. I highlighted here compact in yellow, since the cones of Doyleales mongolica are lax, they are not compact. In 2019, Gunleshi with co-authors described Umcomasia corniculata from the same locality in Mongolia, and in 2022 these authors transferred it in Doyleales corniculata. Thus, this genus Doyleales includes 3 species. Recently, we studied in more detail the epidermal features of both the cupule and cupule stalk of Doyleales mongolica. Here you can see the epidermal cells and stomata of the cupule. In my opinion, these stomata are comparable with those of Jarudia borealis from the New Siberian Islands. Initially, the order Doyleales included only the genus Doyleales. In 2022, Gunleshi with co-authors included 2 more genera, Jarudia, and Tivshingovia in this order. In 2023, Elgoreche and Atkinson restudied the material from the Low Jurassic of Iran, originally described as Carkenia cylindrica. They erected a new genus, Zerabia, with the type species Zerabia cylindrica, and assignment Zerabia to Doyleales. In 2024, Peter Crane with co-authors revised the type species of Dodrictites elongatus, from the Triassic of South Africa. In addition, these authors analyzed similar material from Antarctic, Argentina, and Australia. In the results, they concluded that remains of Dodrictites and the cupules of Doyleales indicate that they are homologous. But unlike other representatives of the Doyleales, bracts have not been found in Dodrictites. This slide shows longitudinal and plan view diagrams of the seed-bearing units of the Doyleales and Dodrictites. I have taken this picture from the paper by Peter Crane with co-authors, and I added 2 new species of Jarudia borealis and Jarudia species. And also I added an apical protrusion here for the Jarudia, since we have found this protrusion in some cupules of Jarudia borealis. According to the Peter Crane with co-authors, the cupule of Dodrictites is flattened in a plane perpendicular to the stalk. Differences between Dodrictites and Doyleales include the tetrahedral seeds of Doyleales and the more tightly closed cupule of Dodrictites. But the most significant differences is the very pronounced distal extension of the cupule here. Jarudia, this is Jarudia, differs from Doyleales, this to Doyleales, and from Tivshingovia in that it has unbranched cupule-bearing stalk with a single cupule at its tip. The cupule of Jarudia is formed by the distal wide flattened part of the cupule stalk, 1 median and 2 lateral flaps, whereas in Doyleales and Tivshingovia, each cupule consists of the cupule stalk, this one, and 2 flaps, excluding the central ovule or cupule of the Tivshingovia, which formed by the stack and 3 flaps. In Jarudia, each cupule contains 2 seeds. where the cupels of Doyleales and Divshinkovi enclose a single seed. Here, the diagram of the two diagrams of the seed-bearing units of Xerabia. As you see, Jarudia is most like Xerabia in having an unbranched cupule-bearing axis that terminates in a single incurved cupule. However, cuples of Xerabia are inferred to contain a single seed rather than two seeds, as known to occur in cuples of Jarudia. Unlike Jarudia, the cupule of Xerabia consists of the distal expanded region of the cupule stalk, plus a distal entire or bilobed flap. I have drawn here bilobed flap. 2 days ago, Evgenia Bugdaeva wrote to me about a preprint of a paper by Lee with co-authors. These authors identified Jarudia and Doyleales from the upturn of South Korea. Thus, the reliable finds of Jarudia are known to date from the late Cretaceous of Inner Mongolia in China, from the Selyap River basin in northeast Russia, and the new Siberian islands in Russian Arctic. Incompletely studied and unpublished material are known from the Transbaikal region in Russia, and there is also a fragment south from South Korea identified as Jarudia, which in my opinion does not have enough feature to accurately identify it, this one. These maps show all known finds of representatives of the Doyleales. Green squares, green because it is Cretaceous, green squares show to the localities of Doyleales in Canada and in central Mongolia. Green triangle shows to the locality of Divshinkovi in Mongolia. Green circles show to the localities of Jarudia, Inner Mongolia in China, northeastern Russia, and Arctic Russia. Blue star shows to the locality of Xerabia in Iran. And also I added unpublished finds from Transbaikalia here, and questionable finds in Korea here. And conclusions. Anatomical features of the cupules and the seeds of Jarudia joy are well preserved, but cuticle details and the structure of the megaspore membrane are unknown. The different preservation of Jarudia borealis allowed epidermal features of the cupules and axis and the structure of the seed megaspore membrane of Jarudia to be described for the first time. The finds in Russian Arctic and northeastern Russia are possibly in Transbaikalia add to the diversity and geographic distribution of Jarudia and show that it grew at a wide range of paleolatitude. Recognition of the new finds of Jarudia reveals a widespread North Hemisphere distribution of this genus in the early Cretaceous and a widespread world distribution of the order Doyleales in Mesozoic. Thank you for your attention.
Thank you, Natalia, for so brilliantly illustrated talk with many new results. Now time for questions. If you like to ask them as it is, just raise your hand, switch on your phone, and I let you speak. Or if you prefer, write your question in the chat. I see Mitya.
Okay, thank you very much for this interesting talk. And the subject is exciting, I have a question from, I'm sorry for my ignorance. So in the past, some members of Doyleales were considered members of Umcomasia, of Corystosperms. And so if you look at Anderson et al. Book, you'll see that Corystosperms, or Umcomasia are limited to Triassic. So those new members of Umcomasia described from post-Triassic deposits are now in Doyleales. So my question is there any robust evidence of the presence of Umcomasialis post-Triassic after the removal of some members to Doyleales?
I'm afraid the same questions. I am not preparing to consider this issue in more detail than I have shown in my presentations. I am studying only one genus, Jarudia, but I know that Gunleshi with co-authors suggested that the cupules of Doyleales, Tivshingove, and Jarudia are homologous to the Triassic Corystosperms, to the Umcomasia included. And they gave a very detailed analysis of the position of Doyleales, and they compare Doyleales with Umcomasia. And firstly, they described as this remains as Umcomasia.
This I understand, yes. But when you remove some members of Umcomasia to Doyleales, do you remove all Umcomasias to Doyleales, or there are some remaining ones from Jurassic, from Cretaceous that still belong to Umcomasia?
As I understand, only Cretaceous Umcomasia were transferred to Doyleales.
Okay, so Jurassic are still Umcomasias?
No, as for Jurassic, I don't know. I only one Jurassic, but it was described as Carcania, and now this is in Doyleales, but the new genus Zerabia. But I don't remember any Jurassic Umcomasia.
Okay. So in your opinion, the order Umcomasialis, or the family Umcomasiaceae, is restricted to Triassic?
I'm not sure in this one, but my colleagues, my co-authors, Peter and Gunle, they named our material as Doyleoid Corystosperms.
So would it be comfortable to ask what Professor Crane thinks of that question?
Yeah. Could you comment, Peter?
Yes. First of all, I should say a really beautiful presentation by Natasha. It's very, very nice, careful work, beautifully done. On the question of the relationship between these Doyleoid plants and, let's call them, the traditional Triassic Corystosperms, I have looked at I visited South Africa and looked at the Triassic, really nice Triassic specimens that John and Heidi Anderson described, which I think are the nicest specimens of Triassic Corystosperms. When we first described the material from Mongolia, we assigned it to Umcomasia because the structure of the cupules is really very, very similar. And you can see this very nicely, even in the compression material from the Triassic. The difference, however, is that the Triassic cupules are not born in these cones. The equivalent, in the material that we've just been hearing about, the lateral units have maybe one cupule, sometimes two cupules, in the Tevshingovia 3, very kind of condensed cupules. But in the Triassic material, the cupule-bearing units are greatly branched and much more ramified. But they still do occur in the axils of bracts. This is very clear. So I think based on the cupules, these plants are very closely related, but on the way the cupules are born, the units are also born in the axils of bracts, but instead of just 1, 2, or 3 cupules, as we see in the Jurassic and mainly Cretaceous material, in the South African material, there are lots of cupules in these axial units, and the axial unit is very branched. So that's my feeling about this. I think these plants are closely related based on their reproductive structures, but of course the leaves are very different, and I think we're still struggling to understand the relationships of this group of seed plants, but I'm quite confident that the Triassic Christosperms and Doyleales have very, very similar cupules.
Mitya, have you satisfied?
Thank you.
Well, we have several questions and comments in the chat. Natalia, could you look in the chat? And I'll read the first one from Barbara Cariglino. Excellent talk, thank you. I'm impressed with the micro CT images. What equipment do you use and how do you set up the specimens? Do you put both part and counterpart at the same time or separately? Thank you.
Yes, we studied separately every specimen's part and counterpart. We studied separately in my tomography. What about equipment? It's not our tomography. It is in another institute, and I'm not competent in the structure of tomography, fortunately. I'm only to give to engineer my specimen, and he give me the files.
And the result is excellent anyway.
Yeah.
And I'm sure you write down in your articles the type of the machine.
Yeah, but now I don't remember the type of machine. Maybe I can look.
Okay, the next comment is from Evgeny Karasov. Thanks for the great talk. By the way, in our laboratory, we have a collection from Transbaikalia, from Chernovsky Kopi locality, where Jarudia may be found. Welcome to Moscow, and we can share it.
Thank you very much. Of course. I will be glad to see this collection too. Yeah.
And a comment from Gwinar. Good afternoon. Is this beautiful material suitable for transmission electron comparisons?
I'm afraid that it is not so good preservation for transmission electron microscope. Yeah.
Pity.
Yes.
Well, a comment from Barbara. Thank you. There is a work by Feng and co-authors that try pteridospermous fructification form the CB, I think, and they set up the specimen together, obtaining very cool results. It is a paper from 2015, I believe. Part and counterpart together, if I understood correctly the comment. Well, Svetlana Popova write down the type of the
Neoscan. Yeah.
Neoscan.
Neoscan. Tomography named Neoscan.
Well, Barbara, sorry for the bad typing, and we're done for now with comments from the chat. If anybody want to ask anything in a more usual way like this, you're welcome to ask. Well, meantime, may I ask a question? What about pollen grains of this group, Doyleales? Is anything is known about their pollen?
Yes, as I remember, Stokey and Rothwell have found pollen grains with two sacs inside the pollen chamber of one seed of Doyleales tetrahedrosperma. Also, as I remember, she with co-authors described one pollen in the integument of the seed of Jarudia joy, and Doyle mongolica are closely associated with pollen grains with two sacs. And in the New Siberian Islands, there are a lot of pollen of the like Alisporitus type with two sacs also are dominated in Paleoflora.
Thank you. So always bisaccate as in Charistosperms.
Yeah.
Thank you. Do we have more questions to Natalia? Peter, comments?
Yes, I could just say that, yes, I think these plants have bisaccate pollen grains, as Natalia says, and as you said. One important question, though, is what are the pollen organs of the Doyleales like? And we did not make very much progress with this in our Mongolian material. So I think this is a big question for the future. I rather suspect that they will look a little like conifer pollen cones, and so may be quite difficult to
recognize,
to associate. But of course, from the Charistosperms in the Triassic, we have good pollen organs. Pterucus is pretty well known. But the pollen organs of the Doyleales and so on, they may look rather different, just the same as the ovulate structures are more compact. That might also be the case with the pollen cones as well. But I think this is a very interesting question for the future that you may be able to solve with the beautiful material that you have available to you and the wonderful localities.
Thank you. Let's hope for the future.
Yes. I think now we can start with look for its leaves and wood. We have some clue about the leaves. The leaves of this group of plants might be ginkgo-like.
Yeah, maybe.
It would be interesting to imagine the whole plant.
Yeah.
And I see one more question in the chat. Natalia. Arun Joshi asking, congratulations for such a nice presentation. What are the prominent features of megaspores of this specimen? Perhaps megaspore membrane is mentioned.
Yeah, membrane, maybe membrane, megaspore membrane. Oh, it's... I have found this one. Yeah. About this one, about megaspore membrane.
I think so.
Yes, but in whole, this megaspore membrane...
Is there the pattern ginkgo-like? What do you think?
Yeah. This megaspore membrane is very similar to the Ginkgoales megaspore membrane, maybe. Maybe only this is a free part of the ends, not so characteristic of the ginkgo, maybe. And so
Thank you. Well, a question from Haru Nishida. Excellent specimens and comparative works. I'm very much impressed. Recent studies widened our knowledge on late Mesozoic diversity of gymnosperms in Eastern Asia and other global localities. There is no comparable specimen from Hokkaido late Cretaceous concretions and other early materials from Japan. I will continue to search.
Yeah, okay.
Let us hope for more finds. More questions? Well, think about more questions, just 1 or more minutes, and I'll just tell you that we have 2 more talks in the spring semesters, but both are in Russian. So the talks in English will recommence on September. And those who can understand Russian come to listen to the very interesting palynological talks in April. Well, many thanks from Dmitry Sokolov. And if no more questions, thank you. And Arun Joshi also writes, thanks you very much for the answer. And Barbara writes, thanks again. And thank you all for coming.